What we talked about last week was just sort of some introductory material about chromosomes, about the cell, about what we can tell from chromosomes. So chromosomes are little packages of DNA and proteins. And in a normal human body cell, there's 46 of these chromosomes. And we inherit 23 chromosomes from each parent. And these chromosomes have our DNA, which basically spells out how to build us. Okay? Identical twins are identical because they share the same DNA. They got the same chromosomes, and therefore they look identical to each other. So your DNA sort of is like a blueprint to build you. And that information is stored in the chromosomes. We have 22 pairs of chromosomes, and then the set, which are the sex chromosomes, which determine sex. XX for female, XY for male. So now what we're going to move into is talking about how do these chromosomes determine our traits and our characteristics? How are they inherited? And we'll talk about some details. Now what we're going to do really is a simplified version of genetics. Genetics is really very complex. And we're going to be talking about a lot of examples that we're sort of simplifying and saying, well, this version is dominant and this is recessive. But in actuality, in most human traits, it's not so clear, not so cut and dry as we make it sound this year. But you'll get into more advanced genetics and understand more about why that's the case as you move on into high school. So we're studying here genetics, and genetics is really the study of our genes, the study of heredity. And heredity means the passing on of information from parents to the offspring. Think about it. If a disease is hereditary, that means it's one that's passed on through the generations in a family. So the science of genetics as we know it has been around for more than 100 years. And one person is very instrumental in helping scientists to sort of understand how traits are inherited is this gentleman. This is Gregor Mendel. He's sometimes called the father of genetics. He was an Austrian monk, a religious person. But he had hobbies, and one of his hobbies was basically botany, growing plants. And what Mendel did is he did his research with pea plants. In the area around the monastery where he lived, he had huge gardens of peas. And has anyone ever grown peas at your house in the backyard or something? So peas grow on like a vine, basically. They have flowers like all fruits, and so they're easy to grow. They have some very clear, easily identifiable traits. So they provided a good model for him to start to understand how these traits are inherited. Okay? And what he did is he grew thousands of pea plants. And as we know, because we learned about plant reproduction, plants reproduce sexually. They have gametes. The pollen grain and the ovule contain those gametes. And so with plants like the pea plant, you can very easily take a tiny bit of pollen from the male parts of a plant, and you can use a paintbrush and brush it across the stamen to get some pollen on the paintbrush. You can then bring it to the pistil of a female flower, brush some pollen onto that, and basically you could cross-pollinate two plants at will. And that's what he did. He crossed hundreds and hundreds of different parent pea plants, grew the offspring, and observed what the offspring were like. He kept very, very careful notes. He was very organized. And he collected huge amounts of data that he then could analyze and look at, and he started to see some patterns emerging. And the patterns that he saw gave us our basic laws of genetics. It's called Mendellian genetics. So what he concluded in doing all this research is that the traits he was looking at in these pea plants, whether they're tall or short, whether the peas are yellow or green, whether the flowers are white or purple, whether the seed's smooth or wrinkled, these are all things he was looking at. And what he determined is that each of these characteristics was determined by two, he called them factors. Today we would call them genes. They didn't know anything about DNA at this point. Nobody had discovered DNA. So it's early on. And so what Mendel concluded is that each of the parent plants was contributing one of these factors to the offspring. And then which factors those are determine the traits of the offspring. So the gametes that he was using to cross-pollinate these plants, they were carrying these factors, as he called them.
How many chromosomes are in a plant?
It can vary. So chromosome number doesn't tell you anything. Some plants have a lot, some have a few. I don't know about pea plants specifically. It's just sort of a random. It doesn't
I wasn't sure if it was every
it's
one
not
has
informative.
46.
It doesn't indicate anything about the organism really. It just is a number that they've evolved to have. So you take a little paintbrush, you put it, you brush it across the anther, you get some pollen on it. You can then take it to another pea plant, brush it across the stigma, and you've cross-pollinated them. So you can choose. I want a tall pea plant and a short pea plant. I'm going to cross-pollinate them, grow the seeds, and see what I get. Maybe I get a tall plant, maybe not. The same concept applies to humans or any sexually reproducing organism. We have the combine each of those gametes. So we know in humans it's 23 and 23, and the sperm and the egg cell. And they combine during fertilization to form a cell with 46 chromosomes. So today we would call these things genes. Mendel called them factors. And these genes give information about specific traits. The genes are in our DNA, in our chromosomes. And so in our simplified version, what we're going to say is that genes come in two forms. Sometimes one version of the gene, we call it an allele, is dominant. And sometimes a different allele, a different version, is recessive. So we can sort of think about the dominant version, the dominant allele, as sort of the stronger form. And we can think of the recessive allele as sort of a weakened form. So it's the version that might be hidden. When we use symbols to represent the different alleles, we typically use an uppercase letter for the dominant version, the dominant allele. Such as if we're talking about the height of plants. Pea plants actually come in these two varieties. Some pea plants can grow and be tall, others grow and then stop and are relatively short. And whether they're short or tall is determined by a set of genes. And actually the tall allele is the dominant version. So we would use a capital T as a symbol. T for tall, capital because it's the dominant version. So the recessive version we write as a lowercase letter. Now really the letters you use are not that important. For us, we're going to always use the same letter. So we're still going to use a T to represent the short allele. But how are we going to know which one is which? Yeah, we use a lowercase. So the lowercase t would be the allele for a short. So because Mendel told us each pea plant has two of these alleles for each trait, there's three possible combinations you could have. You could have plant that is homozygous dominant. This word, homozygous, means that it has two of the same alleles. So if it's homozygous dominant, it means it has two of the same alleles, it has two big T's. So what do you think that plant is like?
Very tall.
It's tall. Or a plant could be homozygous recessive. What alleles do you think that one has? Has two lowercase t's. What do you think that plant's like? Short. So what combination are we missing? X. We call those heterozygous. What do you think that plant's like?
Mixture of
No.
Takes one
No.
together. Takes the dominant.
What do you think?
It takes the dominant.
Yeah, the dominant allele, in this case is tallness, is the one that actually kind of shows through. So this plant is actually tall. And it's just as tall as this plant.
How would you tell those plants apart?
You would have to do something else, which we maybe will talk about on the next couple slides. Yes, we will. Ask that question and when we get 2 slides from you.
What?
Ask that question when we get 2 slides. Yes. All right, now when we're talking about genetics, we have these terms. Sometimes if we're talking about an organ, we can talk about its phenotype or its genotype. Phenotype is its sort of physical appearance. When this organism has these genes, these alleles, how is that expressed? So we use a word, a description. You can say it's tall, it's short, blonde hair, green eyes. We're describing how the genes are shown through. When we talk about an organism's genotype, we're talking about the actual alleles that it has, the letters we use as symbols, its genetic makeup. So, let's say big T, little t is its genotype, little b, little b. Or we could use words. We could say homozygous dominant, say heterozygous. But we're describing the actual alleles present in that organism. So if we look at this pea plant and this pea plant, compare their phenotypes. The one on the left is tall, the one on the left is short. So we know if it's a tall plant, it could be big T, could be its genotype, homozygous dominant, or it could be heterozygous. And we don't know just by looking at the plant. This is a short plant. If it's a short plant, do we know its genotype? Yeah, because there's only one possibility. Little t, little t.
Little t.
Now, Colin asked the question earlier, how do you know which it is? Can anyone think of an answer to that question? Ella? We could take this plant, we call this a test cross. If we take this plant and we breed it with this plant, what if this plant were big T, homozygous dominant, what would we expect?
A tall plant.
All of the offspring from that cross would be tall. What if it were this? Then we would expect there to be a mixture of some tall plants, but also some short plants. And that's how we can determine. That's called a test cross. It's a good question. Good answer. So, Mendel didn't come up with this tool. A guy named Reginald Punnett did. That's why they're called Punnett Squares. And these little squares that we draw allow us to predict what would happen if two parents produced offspring. So let's say, we'll continue on in our tall and short pea plant examples. So if I have these two parents, what are the scientific terms for this parent? First one. Homozygous. Well, what is it? Dominant. And what's the term for this one? Homozygous recessive. So the way we use these Punnett squares is pretty simple. We take one of the parent genotypes and we put it on the top of the box. I'll start with the first one. We split the two alleles, because that's what happens during meiosis. Those alleles split. And then we take the other parent, place them on the side, little t, little t. And then we just combine to form 4 boxes. So what's going to go in this box? Yeah, big T from the top and a little t from the left side. This one's not very exciting, right? Because they're all going to end up with the same combination. So what is this actually telling us? If we take a homozygous dominant and homozygous recessive pea plant, read them together, they're all going to come out to be tall plants. And that's what Mendel saw. He knew that when he had a pure tall and pure short plant, whenever he did his experiments, the offspring always was a tall pea plant. And this is how he started to develop his ideas about these two factors coming together, in dominance and recessiveness. All right, well, then you guys go ahead and fill this Punnett square in between a heterozygous parent and a homozygous recessive parent. Okay? Oh, I skipped this up here. Once we actually complete our Punnett square, often we will be asked to analyze the probability of having offspring with certain genotypes and phenotypes. Now this one, it's simple. What possible genotypes are the offspring? Just heterozygous. What percent of the offspring will be heterozygous? 100. 4 out of 4, 100%. We could also analyze the phenotypes. In this case, again, it's simple. What would we get? 100% tall. All right, now you guys already did this one. So one parent on top, one parent on the side. Combine in each box. Now, typically, we always write the uppercase letter first. It doesn't really make a difference. It's just kind of the convention that we use. Now we end up with something a little bit more interesting. What are our probabilities of the genotypes? Who could tell me the answer to that using some scientific vocabulary? Go ahead, Colin.
You get heterozygous 50% of the time, and you're going to get homozygous recessive the other 50%.
Yep. And phenotype ratio or phenotype probabilities? Half tall, half short. Okay, go ahead and do the last one. We're crossing two heterozygous pairs. All right. So, Punnett square, one parent on top, one on the side. Jack, what goes in this box?
2 uppercase T's.
Correct. Sienna, what goes in this box?
1 uppercase T and 1 lowercase.
Correct. Jake, how about here?
One uppercase T and one lowercase T.
Correct. Drew, how about down here?
2 lowercase t's.
Yep. All right, so when we cross two heterozygous individuals, we end up with 3 possible genotypes. What are the probabilities here? You see it? Yeah, so 25% of them, 1 out of 4, are homozygous dominant. 2 out of 4, so that's 50%, are heterozygous, and 1 out of 4, 25%, is homozygous recessive. So what about the phenotypes? Ella? Yes, 75% of the offspring would be tall, and 25% would be short. And what Mendel found is that he always was ending up with the same ratios. Whenever he crossed heterozygous pea plants, whether it's for the height of the plant, or the color of the flower, or the color of the pea, he was ending up with this 3-to-1 ratio. And when he crossed heterozygous plants, and that's what allowed him to start to think about 2 factors are interacting here. And one is dominant, one's recessive.
How would you know if they're heterozygous?
Yeah, so he would know they're heterozygous because he would breed the same plants over and over again until every single time they were tall. And so we call those his pure tall plants. Then he did the same to breed pure short plants. And then when he crossed them, he knew there was the only possibility in the offspring was heterozygous. And that's how he created heterozygous plants.
Right?
All right, so let's have you guys do, well, let me just say this. Now, these are probabilities, and probabilities are not guarantees. So if Mendel did this, he crossed this heterozygous tall plant with this homozygous recessive tall plant, and he grew 100 of the seeds, it doesn't mean that 50 are definitely going to be tall and 50 are going to be short. Right? This is a probability. It might be off. It might be 55 and 45 or 60 and 40. Probabilities are not guaranteed. They're just likelihoods. If I have 3 children, let's say I have 4 children, what would you predict the number of boys and girls? 2 and 2. Could somebody have children and have 4 sons, 4 daughters? Yeah, of course. The probability isn't a guarantee. All right, you guys go ahead and let's try a different example. You do this Punnett Square. We're crossing a heterozygous yellow seeded pea plant with a homozygous recessive green seeded pea plant. You do the Punnett Square, you tell me the probabilities of having different genotypes in the field. The key is that when you have certain letters, to make them distinct. Like sometimes like with a Y, it's hard to tell like your uppercase from lowercase. I usually make the lowercase one like a cursive. Right, in the genotypes and phenotypes and the probabilities. Any questions? This is pretty easy. Let's see how you did. One parent on top, one parent on the side. Combine to the boxes of our Punnett Square. Genotype ratio. Zainab, what'd you get?
50%.
They're all Y's.
Uppercase y, lowercase y, 50%. And lowercase y, 50%.
Yeah. Yep, 50% big Y, little y, heterozygous, and 50% homozygous recessive, little y, little y. How about our phenotypes? Pretty simple. Alice, what do you have?
You have a 50%.
Correct. Any questions about Punnett squares or how to use them? All right, we're doing good. Because the last thing I have to talk about is not this. These are actually so these. I'll just tell you quickly. These traits were once used as examples of. So in humans, there are very few traits that follow this pattern of simple two versions, one's dominant, one's recessive. You can't just do a Punnett square with your parents' height and say, oh, I'm going to be tall, I'm going to be short. It doesn't work that way for our height. Height is an example of a polygenic trait. It's not controlled by just one set of genes. It's actually controlled by many. And it interacts with our environment and nutrition, stress, and various other things. So we have very few. So there used to be a list of these things people thought and teachers always used in the classroom as examples. Well, here's something in humans that's just dominant recessive. We have since learned most of these things are not simple dominant recessive traits. But we still sometimes use them, but I'm going to skip it. But one interesting it is true is polydactylism. Having 6 fingers on each hand or 6 toes on each foot is actually a dominant gene in humans. If you're born with one of these alleles for polydactylism, you will have 6 fingers.
You will
On each hand. Animals can have it as well.
Yeah, my cat.
So it is useful in understanding that a dominant trait does not always mean most common. It's not that common for people to have 6 fingers on each hand, but it is a dominant trait. It's just relatively rare in our population.
So
All right.
why does the white finger look behind the normal?
Yeah, I know the second one.
The first two.
Oh, let's start here. Put my picture in here.
Wait, your picture was
Can
there.
anyone see the
Then he says that's my bag. All right, we have to finish with pedigrees. So sometimes when we are looking at the tracing a trait through a family, we might use a pedigree diagram. A pedigree diagram is kind of like a family tree, but you're looking at a specific trait as well as the relationship between individuals. Okay? So when we make a pedigree diagram, we have specific symbols that we use. We use squares to represent males and circles to represent females. And if that individual has whatever trait we are just looking at in the pedigree, if it was the disease hemophilia, when a person has that trait, we would shade in their symbol. Shaded in symbol means they have the trait. So here is a man with the trait. There's a female with the trait. And we connect our shapes using different types of lines. So what do you think this line means? They're together in a way, in a sense. It means, now, not quite that they're married, although sometimes it's shown in that way, but if they're connected in this way, they had children together. Obviously, you don't have to be married to have children. So this represents mating, this connection directly from one symbol to another. What do you think this type of symbol represents? Yeah, that's siblings. This is a brother and a sister. So we can use these symbols to trace a characteristic within a family. So let's look at an example. All right, a man that's colorblind marries a woman that's not colorblind. All right, where are we going to put her symbols? Right next door. What shape? Shaded? No. They have children, so we can connect them with a horizontal line. Where are their children going to go? Yeah, down below. We're going to make a spot for each of their children. All right, we have a boy and 2 girls. None are colorblind. So, the boy, what symbol?
Square.
Shaded?
No.
And the 2 girls? Open circles. That's a very simple pedigree showing the relationships within a family and who has certain traits. This shows 2 generations of individuals. Now these can get quite complex. I did with my 9th graders a huge pedigree of the royal families of Europe, tracing the inheritance of hemophilia through the royal families, because it was a genetic disease that was spread throughout the royal families, and you can look at each. See a number there? That's good. Have you ever done these tests before?
I watched like 25.
Yeah, so they are tests for color blindness. So when a person, most types of color blindness are dominant, they are sex-linked traits. A sex-linked trait is a trait carried on the X chromosome. And sex-linked traits are more common in men than women, because a man only needs to inherit 1 copy of the colorblind allele to be colorblind. For a woman to be colorblind, because it's recessive, she has to inherit 2. So it's easier for a male to inherit 1 than for a female to inherit 2. So yeah, there are different types of color blindness. There's red-green color blindness, in which person has difficulty distinguishing between shades of red and green. Here you see the normal colored pencils, and here is what they would look like to a person with red and green color blindness. So if you compare the reds and the greens, that there's no distinguishing. So the tests for this are looking at these patterns, because people that have difficulty perceiving those differences don't see a number there.
42, 6, 7, 15, 45, 6, 8.
Bro, I can't do it that fast.
13, 26.
What?
Can't do that.
42.
No, it's not an actual one. All right, let's finish one last one. So go ahead, draw this pedigree. You'll get a little bit more practice tomorrow, but let's finish with this one. A widow's peak is when your hair dips to a point at the very top of your hairline. If you know the Munsters? Never saw the show The Munsters? Old school, black and white television series? No? The Munsters? No? Right. All right, want to see it? Bell's going to ring in a minute. See how you did. How do we do? I will do some more complex practice tomorrow or Friday.